You said it's freezing? Yeah, for some reason this room is kept really cold. I don't know if they're gonna store the dead bodies in here or something like that. It wasn't ever warm. Okay. So we are moving on to the third topic. So I kind of loosely divide the course into units. They have, a certain degree of logic in that division. But those units are material-wise fairly similar. Okay. So we about to finish the first one. We're going to talk about the cell. I strongly believe that one of the things that you are interested in when is the exam. By the end of this class, you will have a good idea about it. I will have a good idea and you will have a good idea. Okay? I prefer to move the exam down one day so you take it after the weekend. And actually that's what we should discuss now. So there are two options. One, we're done with the material, we take the exam, like next day. Two, we're done with the material, we start the new one, and the exam is going to be after the weekend. Which one the majority prefers will have to do with a majority. Right away or after the weekend? I don't really get a consensus here. Huh? Okay. So far, let's stick to Thursday, but I will do a little bit of a review of the study guide on tomorrow then. How does that sound? Okay? No, stay. It's usually like an hour and a half maybe ordeal. Okay, and then we're done. We move on to other stuff. The summer course is really packed. Okay, anyway, spontaneous generation, cell theory. So first of all, we not never, but for a long time we didn't really know that life is the cellular. Okay, actually for quite a long time people thought that life appears spontaneously. Like mice appear in a pile of dirty socks. Okay, or flies appear on the piece of rotten meat. Okay, frogs in the mud in the river. Just people didn't know where does the life come from. And I think it was 17th century when Italian scientist Francesco Redi proposed an extremely elegant experiment. Okay, he took two cans. Well, I don't think he had these nice beakers at the time, but he had two containers. Okay, and he put a piece of meat in each container. And one container was open, another one was covered. Okay. Actually, there was a third one that was covered not with a cork, but covered with a piece of cloth. Anyway, in the containers that were not exposed to the flies, there were no maggots. So it was sort of a first blow that if you take a meat and make it protected from flies, there will be no maggots. That was first blow towards so-called spontaneous generation theory, right? Second blow came from none else but the famous Louis Pasteur. He's often considered to be one of the founding fathers of modern microbiology. Pasteur put the last nail in the coffin of spontaneous generation theory with his famous experiment, the flask, which had bent neck. So what he did, took two flasks with that S-shaped neck, filled them with the broth, and boiled them for several hours, I think. Essentially, when he boiled the broth got sterile. Does that make sense? Okay. Then he did a very simple thing. He broke the neck of one of the flasks, and he left another one intact. Why S-shaped neck? All contaminants that come from air got stuck into that bottom part of the neck. Does that make sense? It was kind of a trap for the dust and contaminants. You can actually see it here on that illustration. There's like a little contaminants there. Well, guess what? In a few days, the flask with a broken neck demonstrated the growth. The flask with a curved neck remained sterile. This experiment was done in the end of 19th century, somewhere around 1890. You know what's interesting about that flask with the intact neck? It is in the museum of Louis Pasteur. It is still sterile for more than 100 years. It's kind of impressive, right? So since then we knew that, life doesn't appear from something. Okay? The next question to answer was, what is common among all living things? And I vividly remember, I think it was 10th grade or 9th grade of school, when a biology teacher, really nice biology teacher, asked us, what defines life? And he refuted everything that we told him. We told him living things move. And he gave examples of things that do not move, like trees. They don't really move by themselves. Okay? And we told him living things, reproduce, and told, well, we create robots that make other robots. We said living things evolve, and he said there are robots that can self-learn and create more advanced robots. Essentially, one of the key features of life is that all living things consist of cells. One thing that I want to tell you, when I say key thing of life, all living things consist of cells, you have to understand that science has certain boundaries. All living things that we know of consist of cells. We never went to Mars. Like humans never went to Mars. We didn't go to Jupiter or Saturn, or we didn't see anything that could be considered living outside of our world. So anything on Earth, every life on Earth, is cellular. Unless we see something that is life and doesn't consist of cells, we have the right to say this. Okay, so the first person who saw the cell was Robert Hooke, the British biologist scientist. There was no specialization at that time. They did everything. He looked at the cork, but he didn't recognize the importance of the cell. Later, Schwann, that's the guy who worked a lot on the neuroscience, Remarque and Virchow, I think they were all Germans or Austrian. They actually founded the cell theory. They proposed that everything consists of cells, that one cell can divide into other cells, the daughter cells are the same as the mother cell, and so on and so forth. Does that make sense? And then people started to look inside of the cells. And what they noticed, two fascinating microorganisms, chloroplast and mitochondria. Merezhkovsky was the first to see the similarities between chloroplasts and bacteria. Wallin saw the same for mitochondria. What is the similarity? Did you see the pictures of the cell like in the textbook? Did you see the picture of the cell? Did you? Come on. Nod. Or blink twice for yes and once for no. Okay. What is the similarity? First similarity that kind of obvious between mitochondria and bacteria. Bacteria, mitochondria don't have the cell wall, but wait. Bacteria have what? Cell membrane. How many? 2. What else? How, okay, if you look at the typical picture of mitochondria in the book, how does it look like? It looks like a bean. And bacteria look like a bean. So they have similar shape. You know what else is similar? You can take chloroplast to mitochondria, stick it into the egg, they will replicate by themselves inside of the egg. It's kind of interesting, isn't it? So they're capable of semi-independent replication. Later, I think she was American, Lynn Margulis, suggested the theory of endosymbiosis, which is illustrated right here. Long time ago, I feel like a grandfather at the fireplace telling a fairy tale. Long time ago, something like 3 billion years ago, first eukaryotic organisms appeared. Okay? Those eukaryotes were not really energy efficient. But at some point, some of them acquired mitochondria, which was at the time bacteria capable of aerobic respiration. They acquired this mitochondria as the endosymbiont. Endo means inside, and symbiont means living together. So this bacteria started to live inside of that old eukaryotic cell. Does that make sense? Okay. What was the advantage for the eukaryotic cell? To let bacteria capable of aerobic respiration in. What is the final product of aerobic respiration? ATP. So instead of using lousy fermentation, eukaryotes became capable of generating a lot of ATP. Well, I mean, mitochondria generated a lot of ATP, right? So they became energy efficient. What was the advantage for then bacteria, later mitochondria? What was the advantage for? What was good for that bacteria to actually sit inside of the eukaryotic cell? Come on, 2 major things. Protection, absolutely. Number 1. Huh? Say again? More specific. So one of the things, it was protected from the changes in the environment and food flow. It doesn't have to lurk for the food anymore. Does that make sense? It has a supply of nutrients coming from the larger cell. Okay. Eventually, mitochondria, chloroplast, same stuff. They also came from some bacteria, the photosynthesizing bacteria. Eventually, they lost a lot of their genes. They still have DNA, both mitochondria and chloroplast. Okay, they have ribosomes actually that are similar to bacterial ribosomes. They have DNA that is similar to bacterial DNA. And every time cell divides, mitochondria or chloroplasts divide by binary fission, like bacteria do. Does that make sense? That is endosymbiosis. Mitochondria and chloroplasts are two organelles that were acquired by eukaryotes. It's the result of endosymbiosis. Good? Now, before we wrap it up, I want to tell you other stories. I'm full of that shit, so ask me. I love stories, microbiological stories. Okay, we've got cell theory. We know that life is not generated spontaneously. How do we know that bugs cause disease? In fact, Romans and ancient Greeks, they knew something about it. By something, I mean they knew that things can be infectious. Proof of it? Romans built so-called cloaca maxima. It was the first sewage system in the city. Okay? It was actually it was a sewage system that collected waste and dumped it into the Tiber River on which Rome was built to prevent foodborne, oral fecal transmission of infections. Although, I must say, recent studies of helminthic infection in the remains of Romans, good God, they were stuffed with worms. They were worms were everywhere. So that's one. Second, think about the plague epidemics in Europe. I think it was 14th century when Arabs were trying to capture Venice. What they did, they were just throwing corpses of people who died from plague behind the city walls. Very effective, biological warfare. Okay, and it's perfect. Look, all the gold remains in the perfect shape, all the valuables, just people gone. Okay, but still, we didn't know what it is, so it was some sort of a bad air or whatever. Okay, actually, malaria, the name of the disease, means bad air, mal air in French. Okay, so we didn't know what actually causes those infections. First sneak peeks into the microbes and disease were made by Ignaz Semmelweis, the Hungarian physician working in Vienna, and Joseph Lister, the British surgeon. Semmelweis was the head of the maternity ward in a Vienna hospital. And at that time, it was mid-19th century, giving birth to a baby was a very daring experience. You can see the rates of so-called puerperal fever. In some cases, 30%, think about this, 30% of mothers acquired puerperal fever. What is puerperal fever? Sepsis postpartum. When they had sepsis, bacterial infection in the blood after the delivery. What's the outcome? In most cases, in vast majority, death. So if you read old books, his mother or her mother died at birth. That's probably why. Fever. And Semmelweis could not wrap his head around why it happens at such a huge rate in his hospital. So he started to collect statistics. He noticed one interesting thing, that if delivery was performed by a midwife, mother had less chances. That's the collective numbers. We'll get back to that graph. If delivery was performed by a midwife, mother has less chances to acquire the fever than if delivery was performed by a medical resident. So midwives were better for delivery and residents. I started to look at what was the difference among the daily schedule of midwives and residents. Found one striking difference. Midwives didn't go to one part of the hospital and residents did. Any guesses? Residents regularly visited. Huh? Oh, look, it's 1850 now. Morgue. Residents often went to do autopsy. 1850, no gloves. And they came back from autopsy and delivered the baby. Whatever was on that corpse went directly to the mother. Semmelweis did absolutely brilliant and simple thing. He mandated chlorine hand wash. And the rate of puerperal fever dropped to what? About 5%? Right? It was amazing. It was safe now. All they had to do, but look at this, it's mind-boggling to think that doctors did wash their hands before. That's really mind-boggling. Okay? So he mandated the chlorine wash, and rates went down. Unfortunately, the fate of Semmelweis after that was not that bright. He reported his findings on different scientific conferences, but his findings, and he proposed that those were microbes that caused that disease, but his findings were not really accepted by the scientific community, and that was just not acceptance, it was a furious rejection. So he was presented as a crazy dude, and that actually drove him crazy, and he died in the mental asylum where he was placed by his wife and his best friend. They actually, because he went nuts, okay? He died there in very bad conditions. And since then, the phrase Semmelweis effect describes the situation when scientific community violently rejects the new findings that challenge the status quo. Okay, Lister, on the other hand, was much better shape. Okay, he was a surgeon in England, and he suggested carbolic acid, which is the water solution of phenol, as the treatment for surgical wounds. If you ever took the organic chemistry course, and you ever worked with phenol at the room temperature, it's a solid compound. And if you hold it between your index finger and the thumb, which I did when I was in college, and it starts to melt and it leaves a chemical burn. It's toxic, irritating compound. So spraying water solution of phenol on the wounds doesn't seem like a great idea, but at the time there was no alternative, and it actually reduced the number of hospital infections, the wound infections, dramatically. Lister actually he consulted the appendectomy, the removal of appendix, from one of the British kings before his coronation, and the king actually said that he owes Lister his life. Because at that time, imagine, you cut out the appendix, you either die from peritonitis or from wound infection, and due to Lister's findings, nothing happened. Interesting thing, I didn't put this picture here, but if you would Google Joseph Lister, there will be a picture, the old picture of Lister spraying the wound with a phenol on the surgical patient. If you look closer, there's a guy standing next to the head of the patient, holding the head. Any idea why? No anesthesia. You know that phrase that properly secured patient doesn't really require anesthesia. But imagine this, like amputations went without anesthesia. Pirogov used ether. I think he started to use it during the Crimean War, so it should be 1856. It wasn't abundant. It eventually caught up. So second half of 19th century. It's just, it's pretty amazing. So they were first to introduce the idea that infection can actually lead to the disease in the clinic. And then later, Pasteur and Koch, Pasteur was French, Koch was German, they actually established the theory linking the microbes and the disease. Pasteur showed this for things like rabies, and Koch eventually developed so-called Koch's postulates, which we're going to talk about. We don't talk about it here, we're going to talk later. Koch's postulates essentially provide the guidelines how you link a certain microbe and the disease it may cause. How can you confirm that, I don't know, Staph aureus can cause ear infection? What is the proper scientific procedure? Okay, it's not that straightforward anymore, but it was a huge step at the time. So let me wrap